The world's first pig-human hybrid has been created in a laboratory at the Salk Institute in the United States. Scientists say that this is the first ever successful human-animal hybrid experiment, proving that human cells can thrive inside of an animal host. NationalGeographic.com reports. This biomedical advance has long been a dream and a quandary for scientists hoping to address a critical shortage of donor organs. Every 10 minutes, a person is added to the national waiting list for organ transplants. And every day, 22 people on that list die without the organ they need. What if, rather than relying on a generous donor, you could grow a custom organ inside an animal instead? That's now one step closer to reality, an international team of researchers led by the Salk Institute reports in the journal Cell. The team created what's known scientifically as a chimera, an organism that contains cells from two different species. In the past, human-animal chimeras have been beyond reach. Such experiments are currently ineligible for public funding in the United States. So far, the Salk team has relied on private donors for the Chimera project. Public opinion, too, has hampered the creation of organisms that are part human, part animal. But for lead study author Jun Wu of the Salk Institute, we need only look to mythical chimeras, like the human-bird hybrids we know as angels, for a different perspective. In ancient civilizations, chimeras were associated with God, he says, and our ancestors thought the chimeric form can guard humans. In a sense, that's what the team hopes human-animal hybrids will one day do. Building a chimera. There are two ways to make a chimera. The first is to introduce the organs of one animal into another, a risky proposition, because the host's immune system may cause the organ to be rejected. The other method is to begin at the embryonic level, introducing one animal's cells into the embryo of another and letting them grow together into a hybrid. It sounds weird, but it's an ingenious way to eventually solve a number of vexing biological problems with lab-grown organs. When scientists discovered stem cells, the master cells that can produce any kind of body tissue, they seemed to contain infinite scientific promise. But convincing those cells to grow into the right kinds of tissues and organs is difficult. Cells must survive in petri dishes. Scientists have to use scaffolds to make sure the organs grow into the right shapes. And often, patients must undergo painful and invasive procedures to harvest the tissues needed to kick off the process.